Welcome friend, it's Avinash Behra here, and the topic is today is your membrane structure, their function and chemistry. The book I have been using right here is your Baker's The Wall of Cell, probably the 7th edition. Okay, so nothing is important still. Alright, so this is the basic functions, that is your boundary, then the organization of localization of the different organelles, transportations, and your signal detection, and the cell to cell interaction. It is the pipe function of the cell's membrane. And this is the discovery history. Not discovery, this is the history about the membrane structure. Nothing too important. And here one thing I'll show you, this is lipid wrapped. This is enriched with your cholesterol region with that of the protein structure. I think it's so productive. Okay, in this table you can see here the myelin sheath of nerve axon. This is the nerve fiber membrane of nerve cells, and it can show here it is the 18. The protein is 18 and the lipid is your about 79. That means compared to other types of membrane, the myelin sheath nerve axon has a membrane with the enriched amount of lipid rather than protein. And the carbohydrate is also very low in amount, and the protein lipid ratio comes under 0.23, which is far less than other membrane type. Then the membrane typically has some kind of like this structure, that is your carbohydrate chain, then phospholipid bilayer, lipid anchor membrane protein, and integral membrane protein, etc. This is not all important. Okay, then the lipid type, the type of lipid present inside the inside your, what we call this, inside your membrane is your phospholipid. That means a phosphate group will attach with that of the lipid molecule. Another one is sphingolipid. That means instead of glycerol, as in case of like phospholipid, here is a sphingomyelin, which is part of the sphingolipids. The other one is glycolipid. That means the carbohydrate part will attach with that of the lipid part. So this could be a cerebroside or maybe gangliosides. Another one is steroids. Sterols, what do you call sterols, like cholesterol, triglycerol, this kind of sterols. This is a type of structure of your phosphatidylcholine. It is your schematic representation diagram, schematic representation. It is cerebrosides and gangliosides here. It is your galactocerebrosides, and here is your cerebrosides. This graph shows the amount of, that's not important, but the maximum, the phosphatidylcholine are in higher amount. We have mostly the phosphatidylcholine. Let me show you. Here you can see this is a type of phospholipid found inside of the membrane. The phosphatidylcholine, then phosphatidylcholine, phosphatidylserine. These three are mainly found on the outer region, mostly in much amount, where your phosphatidyl inositol and sphingomyelin, this is found mostly in the inner side of the membrane. And during apoptosis, they change their concentration opposite to the present. That is before the present. Then anything like okay, so this is the thin layer chromatography plate, which is being utilized here to separate those different type of lipid present inside the membrane. And as you can see here, the substances which are more polar are being situated towards the bottom, that means towards the solvent system, where those which are less polar, that means they are like cholesterol, which have been moved farther near to the solvent front region. So this is because the acid which is being utilized during this TLC plate preparation, that acid is your polar in nature. So anything which is polar will be get attached and stick towards that acid of that TLC plate, and thus it cannot move upward with that of the solvent front. So the cholesterol is being less polar, will be the maximum less polarity will move farther from this bottom region. Then it's the phosphatidylcholine, then choline, then serine. And anything else? Okay, this is typical membranes with phospholipid, and so and here it's shown that the different kinds of movement being carried out by these phospholipids. The one you can see here in the first, that is your from up to low. That means it is your like transverse diffusion, like flip-flop, and the enzyme being utilized here is your flippage. We'll show you. Then another one type of movement is your rotation movement, another one is lateral diffusion. Okay. The phospholipid translocator is the flippage, which causes the translocation during the transverse diffusion. This is your flippage. And this is the typical phospholipid structure, which is straight, saturated, where unsaturated are being little bit curved, bent structure that cause them more fluid because they will less compact to each other. They arranged less compactly as compared to the other saturated fatty acids. I think I am saying a little bit speed, so I'll try to make slow. Okay, so here's the another thing is here the transition temperature. The transition temperature Tm at which it becomes a fluid. That means a phospholipid or lipid structure of the membrane has some, to being functional, the membrane, it must be in the stage of your, it must be maintain the fluidity, and to maintain this fluidity we need amount of cholesterols to be get actively work together. So whatever may be, the transition temperature shows that it is the change in your phase of that lipid membrane structure. That means from gel phase to fluid phase and fluid phase to gel phase. So this is called a phase transition, and the temperature which regulates is called a transition temperature. We'll discuss coming slide. Here another one thing, your fluorescence recovery after photo bleaching. That means the membrane is being tagged with fluorescence, then the light caused the bleaching, that means they fade their color, and then because of the lateral diffusion of the protein, the region becomes once again filled with the fluorescence tagged lipid molecules. So the graph will show like in this way. Hope you understand, or you can comment below. Here is your lateral diffusion, the same thing being experimentally done here, and so on. Okay, nothing so important new. Okay, so here's the transition temperature I'm talking about. You can see here is the heat absorption by this lipid molecule at the temperature of 28 degrees Celsius. You can see below this temperature, the phase of this lipid molecules to become a gel phase. That means it will be less functional as compared to the more temperature region, that is the fluid phase of the lipid molecule, which is actually as compared to it's comparatively more fluidity and more functional. So this is your normal membrane with temperature Tm. Okay, let's come to the what we call the more unsaturated fluid molecule, another saturated less fluid molecule. As I have shown earlier figure, that those lipid molecules which are saturated, and they are very compactly arranged together, which makes them more condensed and less fluidity. And this is where the transition temperature of this kind of saturated phospholipids has a comparatively higher transition temperature as comparable to that of your unsaturated fatty acids. So the more unsaturation in fatty acids in the membrane, the transition temperature will be definitely lower, and the other one will be obviously the higher. So here another graph is being shown here about the number of carbon atoms, because we have discussed here the saturation, that means the double bond and single bond temperature affecting the temperature, affecting the phase of a lipid molecule. And here it is being described, the figure shows here that the number of carbon atoms, how they regulate the transition temperature. Because as you can see here, the orange bar right over here is shown that effect of chain length on melting point, and the second one is effect of unsaturation on the melting point. The more the unsaturation, the lower, you can see the transition temperature will be lower. So the number of your double bond increases, that will be definitely decreases the transition temperature, and because it will be less compactly arranged, that's why. And when you increase the number of carbon atoms, the transition temperature also increases, because in that condition also they will be more compactly arranged. So the one thing that I am to tell you, that the two important point about the transition temperature is that one, that it must be unsaturated to becomes lower transition temperature, and second one is it must be a low carbon atom chain length. Yes, it must be a lower carbon atom chain length to become less transition temperature. I mean to becomes lower transition temperature. Okay. This is a figure of a compact arranged, and this is not compact arranged C strand contribution, and this is typical phospholipid structure. Nothing so important here. This is also a typical membrane structure. Okay, so this is, you can see here the types of arrangement of different protein molecules inside the lipid bilayer. So the first one you see in the integral monotropic protein, that means attached towards only inner or outer surfaces. And the second one is your a single pass protein, but that means a single transparent protein is there. A multi-pass protein means the single protein molecule being multiple time passed through the membrane. Second one is a multi-subunit protein. That means different types of protein or multiple subunits are get together and get passed through this membrane. Second one is your peripheral membrane protein attached to that transmembrane protein, not to the lipid molecules. Lipid bilayer. Then another one is your fatty acid isopropyl alcohols. Then we attach with an alcohol group rather than that of the directly attached without attaching with that of the lipid molecules. Yes, it's attached through the fatty acid. Yes, towards the alcohol and fatty acid ester bond. Okay. The second one is a GP anchored protein. And this is your glycosyl phosphatidyl inositol. Yes, this is GPI, glycosyl phosphatidyl inositol phosphate anchor protein. That's the important one, but I cannot express that. Okay, next one is your not so important, but the structure of two integral protein. You can see here how the pass through they are function like something. Let me check anything important. Okay, this is your SDS electrophoresis chamber. You can see the SDS being attached towards the protein to get them all surface with negatively charged molecules so that they can move towards the positive side anode, and thus they can be separated and be analyzed through your devices, which will be make them visible. Okay, nothing so important. Okay, hydropathy analysis is one of the most important thing you must be learn. Here in this book, it's not correct. Not much said about the hydropathy analysis, but this is a technique to analyze the different types of proteins, their structure without undergoing the X-ray crystallography. That's why it's the advantages over the X-ray crystallography. This hydropathy plot shows considering the charge of the protein, the structure, the contribution towards the structure of the protein. It mainly includes hydrophobic amino acid types of proteins. So this is the basic idea. So you can go through the videos other than diametric videos to get more knowledge about this hydropathy analysis, and you must definitely go that. Anything else? Let me check this out. Not so important. No. Okay, this is a little bit important, not that much important, but you can see here this is the typical membrane structure with mostly the bilayer, and you can see there are the three different type of proteins being shown here. The outer one is peripheral protein, and in the peripheral protein another one is your transmembrane protein. So when we need to tag those protein molecules, we need to be tagged with some what do you call tagging substances, and to tag the substances we need some enzyme. So we have to put that enzyme. So in the very first thing you can see here, the lactoperoxidase is being enzyme being utilized with that at the iodine 125, radioactive, obviously radio labeled, to tag with that A and B protein. But what if you need to tag only the inner side of the membrane? Then we need to first put that membrane in a hypertonic medium. That means the surrounding is being hypertonic as compared to that of the inside, so that the enzyme could be get inside, and after it get inside, then we can remove it and put it once again in isotonic solutions where the charged molecules could be easily move and come all around, and then will be tagged by means of that lactoperoxidase enzyme, and then it will be undergo microscopic studies. Anything else? Okay, this is a very not that interesting, but you can see here the different this is your mouse cell and human cell membrane being taken here, and then it being exposed to a fluorescent antibodies, and those fluorescent antibodies are very specific towards the protein, different proteins present inside a mouse and a human cell. So after some time it found that the cells get fused, and this will be observed through fluorescence microscopy. Nothing.